So today we're going to talk about different plant cell types and specifically reproduction. So this builds on last week. We're hoping to describe specific cell types, and this references your book and especially their functions. So we'll be talking about things like xylem and phloem and how these things might be affected by hormones. We'll also talk about comparing and contrasting mitosis and meiosis. This is especially relevant to reproduction. And lastly, we'll discuss artificial selection of plants, and talk a little bit about, what are the products and the how-tos of doing artificial selection. So those are the goals for today. So in reference to plant cells, there's some different types. These reference to texts and books is table 10.1. Some of these we already mentioned, maybe a little bit in class. But there's epidermis, which is the outer covering of the plant, meristem, which is the shoots, the baby cells, the cortex, which is fills most of it. The xylem and phloem, which we'll talk about, specifically parenchyma and parenchyma, are all the main types of tissues in plants. So you might want to know about what is meiosis. You might recall from biology that meiosis is a process that generates gametes. So this would be sperm, egg, pollen, and so forth. The way you could describe gametes are that they are haploid. They only have one copy or one end cells. The steps that occur during meiosis, there are two cellular divisions, and they follow a process called meiosis. And remember, starting with prophase, metaphase, anaphase, telophase, and cytokinesis, which occurs last. There are again two cellular divisions. The big focus we have is recombination of homologous chromosomes. You can mix up traits to generate something new. And that's the big thing about meiosis and sexual reproduction, is it creates new things. So we're going to talk about what happens in meiosis. We start off with a 2N and 2C cell. This is our starting point. Up here, your generic cell. From there, we are able to copy things. We use the S phase to make a DNA copy of everything. And that doubles the C value, which refers to the DNA content, versus the N value, which determines whether it is diploid or haploid. How many copies from the mother or the father does it have? Is it haploid, meaning only one copy or one set, or is it diploid, meaning two? As we move through this diagram, we see that we were able to copy 2N4C. That divides into a 1N, so now it's haploid. And since we divided it into two cells, 1, 2, the DNA content also divides in half from 4C to 2C. Later we want gametes that only have one copy of DNA, so that's why we split and we go one more division to end up with the 1N, 1C content. From there, we also want to talk about what is mitosis. Mitosis is for your toes. It's general growth and repair. So this is mostly what's going on in your body right now. Anytime a plant gets some scar tissue, it's going to go through mitosis. Some key points: mitosis has only one cell division. It still follows meiosis, but there's no replication for repro. DNA synthesis is just for the creation of two daughter cells. So we're going to wrap this up with some reminders about reproduction. What is artificial selection? Artificial means man-made. So this is man selection. This is usually done to favor specific traits beneficial to the economy, or to create kind of subspecies. An example of that is basically all a lot of the broccoli family, peas, are all modified from an original to emphasize certain traits. The same is true of cauliflower and Brussels sprouts. You decide what traits matter, and you made them. This would be analogous to kind of animal breeding, specifically dogs. Artificial selection, who did it have to do and what did it have to deal with? You might want to think back to a guy named Mendel. Mendel was the guy who came up with everything with the pea plants. But he first started his first work in artificial selection, which had a lot to do with the choosing of the mates and the forcing if they would ever exist together. Some of the products of artificial selection we're talking, I mentioned them earlier, but things like animals, plants, specific traits, size, and the same would be for animals in terms of breeds and work, which is all important to think about. Lastly, how do you do artificial selection? If you think back to the anatomy of the plant, we have the anther, maybe the stigma and the style. The main point is that you are going to collect pollen from here and rub it onto the stigma and style. This will cause the formation of the pollen tube and fertilization. Artificial selection for plants is really easy. It's easy to just grab and choose and select and pick. A lot harder with things like animals, which is more difficult. I'll talk about some of the examples of why that is so crazy, one of them being pandas. Is one of the issues that has to do with artificial selection. So in summary of the lecture, what do we want to hit on? There are different parts of plants pertaining to their functions. We want to relate the idea of structure and function. Again, we want to hit on the differences between differences and similarities between mitosis and meiosis. And lastly, what happens during artificial selection, pertaining to what role do humans play in determining phenotypes and genotypes in the population. So that's a summary of the lecture. If you have any questions, always feel free to give me a call. Shoot me an email. This week we'll continue working on mitosis. Meiosis, and working on plant parts. And then next week we'll hit into Mendel, chi-squared, and counting up all its genetics using math. But again, shoot me a message, give me a call. Otherwise, take care and enjoy your day. Hopefully you learned something about plants.